Good morning, my name is Emilia Jarachowska. I work at Erlangen University, and together with my colleague Niklas, we would like to present a talk that actually does not report new original research data, but instead we would like to stimulate some reflection and some discussion on data handling. And I know you will be like, why are we talking about this again? You are probably constantly hearing about reproducibility and open data, but we think we have a very convincing example to illustrate that huge discoveries in paleobiology can be severely compromised by poor data handling, and it starts in the field. It starts the moment you collect your samples, and then it is also affected by the way you report it. So in this talk we would like to argue for a better standard.
So for the example, I'm going to talk about one section, which is Seymour Island. This is a small island in Antarctica, which is well known since it has a very good preservation of the mass extinction at the Cretaceous Paleogene boundary. And the problem is that it has a very homogeneous lithology and it's hard to reach. So doing fieldwork at this section requires a lot of effort. The data set I'm going to talk about was sampled in field seasons 82 and 84 and was originally published as part of a PhD thesis.
On this map, it is shown where the measured sections are located on Seymour Island. Overall, there were 5 sections measured that are spread out over 4 kilometers of the island, and they have an altitude difference of up to 1,000 meters.
While measuring these sections, fossils were collected in sampling bins between 2 and 39 meters and were noted as being either present, abundant, or very abundant. The problem is that within that PhD thesis, no actual sampling procedure is noted, which makes it hard to reproduce these sampling procedures, since it is unclear how much sampling effort actually goes into each bin. This is amplified by the different size of the sampling bins, since it is not clear whether an abundant species in a 2-meter sampling bin is equivalent to an abundant species in a 30-meter sampling bin. Based on this PhD thesis, multiple articles were published. I will only talk about 2 ones because they are most relevant for the example discussed here. The first one was a publication that only tackled stratigraphy and petrography of Seymour Island. It took all the samples collected during fieldwork and documented grain size distributions along the different sections measured on Seymour Island. It also includes a sequence stratigraphic interpretation of the section, which includes a maximum flooding surface and a high stand right at the Cretaceous Paleogene boundary. The second publication only documents ammonite occurrences on Seymour Island that were collected during these field seasons.
This figure is taken from this publication, and it is actually a central part of this publication. It documents the vertical distribution of ammonites found on Seymour Island. However, there is no reflection of the original sampling procedure in this figure. It is neither clear that this is actually data that has been generated by correlating data across all of the island, nor is it clear that the original sampling was in bins and not continuous, as the mode of representation of this figure suggests. Because of the suggestive character of this figure, this data has been digitalized and taken by more than 10 different authors to derive information of the Cretaceous Paleogene mass extinction. This is taken from one of the publications that works with a digitalization of that figure. It takes every dot in the original figure as one ammonite occurrences and tries to derive information about the mass extinction at the Cretaceous-Paleogene boundary. However, no information about the original sampling procedure is there, no information about the sampling bins is there, and also no information about the potential sequence stratigraphic overprint is mentioned in this publication.
So this example clearly demonstrates the problems that are generated when sampling procedures are not documented properly and it is not traceable how data sets are actually generated. This section in Seymour Island is one of the best sections for documenting the Cretaceous-Paleogene boundary, and there is potentially a sequence stratigraphic overprint of it, which will strongly alter our interpretation of the Cretaceous-Paleogene boundary. And due to the improper documentation, we are actually not even able to tell whether there is a sequence stratigraphic overprint. This all really emphasizes that we need to create a sampling procedure that tightly fuses sedimentology and paleontology, because otherwise our data sets are not usable for modern paleobiological work.
This one extreme example illustrates a problem that is very widespread in the fields of paleontology and paleobiology. And in this talk, we would like to review some reasons for this and look for possible solutions. And the motivation of our talk is that taxonomic and biostratigraphic expertise is currently undergoing extremely fast erosion, but we need it for our work. And we would like to suggest that better data handling can allow a better use of this data and its higher longevity, on the condition that people who produce this type of data receive credit for their work and they benefit from doing it. So how to make sure that nobody will use your data? First of all, do not give GPS coordinates, nor provide any information on where your sections are located. If you include a map, mark your section on a paleogeographic map of the world. Do not illustrate your specimens. Everybody knows you're the expert. Use regional stratigraphic units, especially if they are topostratigraphic. Basically, you can throw away your copy of geologic timescale. Avoid mentioning details on lithology and do not include any logs of sections. Information on bedding, discontinuities, hiatuses, or lateral variability can only distract. You can see all the talks by Niklas to find out why. If the reviewers insist on sample positions, make sure they are illegible. Use raster graphics because they are hard to magnify or decompose. Do not include a data table. If you do, make sure it is not possible to process it using a computer, for example, save it as a graphic file or use excess formatting. Provide only the age without the data upon which it is based. For example, only the biozone, but not the taxa that it was based on. When calibrations or timescales change, nobody will be able to tinker with your results. Do not cite taxon authors. And do use a mixture of form taxa, subspecies, and open nomenclature, so your data won't be amenable to any diversity analysis. This list is made based on our experience with genuine studies published recently and in leading journals. The question is, why are people not motivated to document their data? How can they be motivated? Obviously, they are not rewarded sufficiently for making this extra effort. So how can they be rewarded? A commonly cited argument can be summarized in the following quote. In most cases, the scientist is the custodian of data either owned by the employer organization or, if the data collection was paid for with public funds, then the data should belong to society. Unfortunately, Some scientists believe they personally own data, even though their salaries and research are funded by the employer or government sources. Most scientists are stewards of public data and have a responsibility for its dissemination. But the reality shows that researchers, and especially taxonomists, struggle with being recognized, and the long and tedious training they require is not recognized sufficiently. Preparing data for archiving requires additional work. Moreover, studies show that many scientists are afraid that if they make the data available, someone will misuse it, for example, to support other conclusions than what they think is correct. Finally, many people are afraid that someone will find a mistake in the data or analysis, and, unfortunately, our own experience shows that digging into people's data on and off proves that this fear is justified. But exactly for this reason, better data handling is needed. A commonly proposed solution is rephrasing the problem from data sharing to data publication, where researchers receive credit for correctly archived datasets just as much as they do for research papers. But the infrastructure for implementing this is just budding, and it would have to be actively adapted by funding institutions, journals, and employers. Current publication model does not reward taxonomic or biostratigraphic data collection and documentation. We think that it leads to misuse and overinterpretation of such data, in that authors of biostratigraphic studies are compelled to exaggerate the importance of their study in terms of ecology or evolution, even though these are not topics they addressed and the data has not been collected with these questions in mind. Otherwise, such manuscripts are often rejected as regional, parochial, or narrow in scope. We would like to draw your attention to the initiative of the open access journal PeerJ, which recently offered a prize to reward early career researchers who have shown commitment to rigorous science. We think this is an excellent initiative, and it could be adopted by many paleontological bodies. We will be advancing proposals to introduce such prizes to major organizations. So what can you do now to reward proper sampling and documentation? When hiring, you can consider taxonomic expertise and rigorous documentation. As editors or reviewers, you can require full datasets and review them. You can encourage, solicit, and reward publications with properly documented new taxonomic or biostratigraphic information. And very importantly, you can require citations of all publications which provide the data so that previous work receives sufficient credit. When reviewing grant applications, you can actively support funding for training in taxonomy, fieldwork sampling, and data management. Each of us can make sure that we cite datasets correctly, that we cite all publications which provide taxonomic data, even for large compilations, and that we even cite database contributors who help to make this data digital. Here are just a few examples of archives and databases where paleontological or biodiversity data can be stored, mostly at no fee at all. Some institutions offer support in research data management, but still too few researchers have access to such support at a professional level. There are, however, online resources which you can use to educate yourself or help train someone. The point is to take the burden of managing data away from specialists and give them support so they can focus on scientific work. I am aware